Good afternoon, everyone. I hope all of you have had a nice lunch. It's almost time to start our oral presentation sessions. I'm Varun Kher. I'm a faculty here at the Wildlife Institute of India and your host for this session. Uh, this particular session is titled Innovations and Interventions in Wildlife Conservation Science, and it will have a total of 6 presentations of 10 minutes each. For the presenters, we just have some housekeeping announcements. At 5 minutes into your presentation, there will be a yellow colored placard that will be displayed on the left side here. When there are 2 minutes left, there will be a red colored placard, and the placard will be waved at the end of your time. It is requested that everyone keeps to their time. I'm pleased to announce the first speaker for this session. It is Laura Bertola. Laura is a research associate at NCBS, and she is interested in utilizing genomics for biodiversity conservation. For a PhD from Leiden University, she explored the genetic diversity in lions and has worked in population genomics of many African and Asian species. I request Laura to please come and start the session.
Should I open the... Okay, thank you very much. I'm very happy to be here today and present some of the work that I've been doing and trying to give a bit of a broader overview. And I'm sure you're all are very well aware that with the recent increase in sequencing capacity and prices going down for genomics work, there's more and more big-scale genomic data sets coming out also for non-model organisms such as wildlife species. And of course, these data sets have an enormous potential also to be informative for conservation decisions, but that's sometimes not fully used to its full capacity yet, is my impression. So before joining NCBS, I spent 3 years at Copenhagen University, where there's a big program on large African mammals, and so these are all papers that came out from the time there on various species. Basically exploring the distribution of their genetic diversity across the entire continent or their entire range. And even though often conservation was given as a motivation for this kind of work, it was often kind of more of an afterthought, because to be honest, a lot of these papers are more about evolutionary genetics, about speciation and gene flow and divergence times, and not so much directly relevant for on-the-ground conservation decisions. However, I want to make the case here that these data sets are actually enormously useful and can be used for conservation. And I want to illustrate that a little bit more with a species that I've worked on for quite a long time now, and that is the lion. So when I first started working on lions, I was interested in the distribution of genetic diversity, but across all the different populations. So this became a hugely collaborative work where we teamed up with a lot of colleagues from different countries to collect samples and get some insights in all of these different populations. And we were kind of opportunistic in our approach. In some cases, we collaborated with people who were putting GPS collars on lions, and we could get blood or tissue samples. We also had field teams on the ground that collected non-invasive samples like scat and hair samples. And we also leveraged natural history collections and sampled skulls and old skins to kind of fill the geographic gaps to get a nice overview of genetic diversity or genetic variation in the lion. And we did a bunch of different things. So we started out with looking at mitochondrial DNA, and we did microsatellites. Then we did some whole genome sequencing. Based on those data, we designed the SNP panel. We then did genotyping with the SNPs. But I always try to go to meetings where there's also a lot of managers and policymakers present. So I try to not only speak to my fellow scientists at scientific conferences, but I actually also speak to the people who actually are involved in the decision making. And they always kept highlighting that the problem is, first of all, they often don't have access to the scientific literature. A lot of it is behind paywalls. And a lot of this information is so fragmented, because before I started working on lions, other people had worked on lions, and they had published their data sets on a different set of microsatellites or a different mitochondrial gene. So the information is very fragmented. And so on one of these meetings, we decided it would be great if we can collate all of this information and kind of write a paper on kind of like a baseline data set for genetic variation in lions, and also genetic considerations specifically for long-distance translocations, because these are quite common on the African continent, not just for lions.